All right, so now we're going to describe part one of the digestion lab. Here, the salivary digestion of carbohydrates. We're going to be using this saliva that was generously provided by my lab mate. And this will be the enzyme. And then we've got, our substrate for the enzyme, our starch here. And then we can look at how this process happens under different conditions. So to start, we're going to have to just put some starch. We have 3 mils of starch that we put into each of these 3 tubes. I'm just going to bring this up here. I'm going to go, there's 3 mils in there. 3 mils in there. 3 mils in there. And then also 4 and 5 are going to be controls. Part of an experiment, whenever you're using some kind of an assay, you want to validate your assay. Make sure you give your assay something where you know what the answer is supposed to be, and make sure you get the answer that you're expecting. So then you trust what the answers are when you're looking at your unknowns. So for test tubes 4 and 5, we're going to have test tube 4, which is just going to have pure starch in it. And when we do our test, it better say that, oh, it just has starch in it. Test tube 5 is where we're going to have pure maltose. I might as well just put that in right now. Well, I will put that in a little later. And so when we can check our assay for maltose, that better say pure maltose with no starch. All right, so we have starch in our 3 unknown tubes here. Test tube 1 is going to just fill out our volume by adding 3 mils of water. So we can go 1. Have to make sure I can see this thing. 3 mils of water. So we got 3 mils of water in test tube 1. Test tube 2, we have to add 3 mils of this quality high grade saliva. And I'm just going to bring this up to 7 so we spit it out. I'm going to spit it out into there. For test tube 3, before we add the saliva in, we are going to increase the pH by using this concentrated hydrochloric acid. This stuff is something you got to be careful with. You should not be using this without wearing eye protection. Regular glasses can work. If you don't wear glasses, you got to be wearing goggles of some sort. This can damage your eyes, and your skin or your clothes or anything. So be careful with it. We're gonna add 5 drops of the concentrated hydrochloric acid here. 1, 2, 3, 4, 5. And make sure, so now we have this very low pH solution, and now we're going to add 3 mils of our saliva into this. So 3 mils of saliva, but the conditions here are going to be very acidic for this salivary amylase to try to do its job. It's important to remember, keep it in your mind now, that each of these tubes we're going to be testing both for starch using iodine, as well as testing for the presence of the products of digestion, maltose, which we're going to use Benedict's reagent for. So we're going to be putting these in the incubator. And after we're done, though, we need to make sure we take each of these tubes and we split it into 2 tubes. That way we can do the Lugol's test to look for starch in one set, and we can use the Benedict's test on the other side to test for the presence of maltose. If you forget to do this and all of a sudden put your Lugol's iodine in here without having split it ahead of time, then you're not going to be able to do the test for maltose. So at this point, we're going to take our tubes here and we're going to have to incubate them at body temperature. And we'll come back in a while and see what happens. So here we see test tubes 3, 2, and 1 are joining the test tubes from parts 2 and parts 3. And we're going to close this puppy up and come back in a half an hour or so and see what happened. Alright, so these have been incubating at 37 degrees C, and now we can do our assay and see what the results are. Alright, so each of these needs to be split into 2 roughly equal test tubes so we can check each sample both for maltose as well as for starch. And again, it doesn't matter exactly, it should just be roughly equal. And again, these are my 3 experimental tubes here. And then remember, I also have the control tubes I have here. This is just pure, 4 was pure starch. So when we do our test, this should show up positive for starch and should not show any maltose. This one here is pure maltose. So when we do the test for maltose, this should be positive for maltose but have no starch present. That's how we're going to know our test is trustworthy. So we can start with the test for starch. We use this iodine, and how many drops do they say? 3 drops of New Logos iodine, so I'm going to put 3 drops in each of these tubes. 1, 2, 3. 1, 2, 3. 1, 2, 3. Whoa, that's not good. Oh, that's good. 1, 2, 3. And 1, 2, 3. All right, so if we look for starch, 1 is dark purple black, presence of starch. 2, this amber, that's just the color of the iodine. So this would be no starch, looks like. 3, we also have a very strong reaction here. And this is encouraging. Our pure starch is showing positive and pure maltose is showing negative. To do the maltose test, we have to add in 4 mils of Benedict's solution into each of our other tubes. So now I'm going to, again, for each tube, let's clean this up. And we'll get, there's 4 in there, 4 in there, and finally, 4 in test tube 1. All right, so now each of these is going to have to go and get boiled for 2 minutes. So let's put these in. We have this in boiling water here. We could also make sure, like when you're using a Bunsen burner, make sure you have a nice blue flame. That's hot. If it's not adjusted, you know, this is not hot. The water's not going to boil. So you want to make sure you have the blue cone of heat. And probably lower this. No, we're not going to. So again, just to show you again with the starch, pure starch, is going to have this very strong reaction where you get this purple-black. And in the case of the maltose, where there was no starch present, we can see the iodine just stays this kind of amber color. So this was no starch at all. This is showing presence of starch. And we'll come back in a couple of minutes when the Benedict's solution has had a chance to do its magic. All right, so these have had their 2 minutes in the boiling water. I'm going to carefully put them back in the rack here so we can see what happened. There's our controls. There's tube is this? This is tube 3. And tube 1. And here we can turn off the fire. And now let's go take a look. So first we can go to our controls again, and we can see this Benedict's is blue initially. It stays blue if there is no evidence of maltose or any of these reducing sugars. And so this is what we were hoping. This was pure starch in here without any maltose, and this shows from Benedict's that there is no maltose present because it's staying just blue. This would be a very strong positive reaction with Benedict's, where it turns this reddish orange. Again, there's a whole range of colors it can turn. It can turn from kind of greenish all the way to this kind of tomato red orange. That would all imply the presence of maltose in our case here. So this is great from our control perspective. Starch was negative for maltose, maltose positive for maltose. If we look at our unknowns, this was 1. This was starch just with water. So starch basically on its own after sitting at 37 degrees. Blue means, what does it mean? And why? 2, this is showing positive for Benedict's. We definitely got maltose appearing in this tube. We can talk about that in your discussion, why that makes sense. And 3, this one was the same as 2. These were both exactly the same, except in 3 we had that concentrated hydrochloric acid in ahead of schedule before we put in the amylase. So the enzyme ended up having to try to do its job under this really low pH condition, and we can see a very different result. Definitely something you want to think about and discuss.